let me give you a brief introduction about Netri. So Netri is a French company. So we're based in Lyon, in the capital of the goal, as we say, the capital of food also. In France, we've been existing since about 6 years. Basically, now we're composed about a team of a little more than 30 people and a lot of science inside with a PhD multidisciplinary team that is conducting all the development that we're doing at Netri. So basically, Netri does everything from research, development, to production, to support, to service, of course, to sales and marketing associated with.
Can you pause a minute? Can you make your presentation to full screen? I think we can see all of your slides right now.
All right, excellent. Is it better?
Yeah, better. Thank you.
All right, thank you so much. So the idea of Netri is really to work on a scalable, high throughput, and easy to use device, organ-on-chip and organoid-on-chip devices to improve the drug discovery process and optimization of drugs, which, as you know, represent a big challenge to the market, to the economy, generally speaking, as 90% of the molecules are not that are discovered are not going to go until the authorization and approbation of the regulatory bodies. So we have decided from the beginning to address this type of devices and these technologies with a production mindset. That means that our production facilities, which is still small at the moment, 200 square meters, and then we're moving to another building actually in 2 months from now, which will be 5 times bigger. All the production that we are doing is done in a very high-class clean room level for this type of devices. And we also have laboratories where we perform drug testing for customers as well. So basically, what we're talking about, this technology, what we have between the 2 fingers of the operator here is a set of 4 chips which are basically identical. This chip, this chip, this chip, and these chips are all identical. They're coming from the same mold, so this is the same architecture. And we're going to discuss now what we are talking about with those organ-on-chip. So as you know, there are already conventional methods to allow the mix of different cells and trying to build a platform, biological platform, based on the central well, which is generally what is doing. The organ-on-chip, generally speaking, has leapfrogged the technology in a sense where we, with the microfluidics approach, whatever higher, the idea is to, start to mitigate the natural compartmentalization of the anatomy. However, there are different approaches, and since it started about 10 years ago with 1 or 2 historical players, they have tried, to be very complex because they wanted to be, thoughtful. And this is where probably the old technologies are not necessarily working well those days. So we took the approach of, as I said, being very simple and also trying to be smart. So from this mold here with the 4 chips, let's go now on 1 chip and explain you what is the technology. Once you have understand the technology, then you can project yourself through the presentation and the different applications and readouts that help to characterize the pharmaceutical drug testing. And not only pharmaceutical, actually, nutrition, and also in the cosmetic and dermatology, for example. So 1 chip here is composed of 6 wells, okay, and of 3 channels. That means that 1 channel has 2 wells. This is the channel 1, if you follow my pointer. This is the channel 2, and this is the channel 3. We have two wells because 1 well, you decide which one will be the inlet where you're going to pipe it in, and introduce the cells and introduce the media to nurture and to feed the cells. And one is going to be the what we call the outlet, where you can basically pipette out. The idea of the technology of Netry is to be able to grow separately in each channel the cell culture that you're looking for. That means there is no mixing of fluids. This is very important to understand. And we have a specific trade secrets as well as technology secrets that help us and know-how, because basically the middle channel here is acting as a fluidical wall, and that's how we maintain the separation of the fluids between the three channels. So number one, you can do co-culture or tree culture in all devices without mixing the fluids. The beauty of our technology is that we add some micro channels which are located between the channel 3 and the channel 2 and between the channel 2 and the channel 1. So at the inter-channel, not in the channel, but between the channels, they are very small micro channels of 3 by 5 micron section that allow only the axons to project through, okay? So if you look at the image here, you see that this is the channel 1 with cell bodies here, and the projection all over when they grow. And also, of course, when the axon reach the interchannel, they use the microchannel. It's like a tunnel under the water. So they take, the axon takes the microchannel, then it arrives to the second channel. At the second channel, then it's free to operate again. In the middle channel, like in the two other channels, it is open. There is no microchannels. However, they will project and go at some point, finish their journey in the interchannel between 2 and 3. And that's where the axon will again take the guidance of these microchannels to reach the channel 3. So illustrated here from a body, then you will have projections and you will have endings, the neurites on this side, okay? So that's one of the key things that you need to grab in our discussion now. The other thing that is unique in our technology is the ability to perform our technology very simply without any equipment. It's a standalone device. Why? Because the PDMS, which is the material we are using, is permeable to gas, so to oxygen, which means that the cells can breathe during their maturity. In other words, when you introduce and you seed the cells, then you need to feed them every second day by changing the media. That's all you need. You don't need pumps, you don't need equipment to let them roll, to let them run. So the static seeding protocol that we have in place is already perfectly working. That's number one. The second thing is the PDMS is transparent, so that helps a lot for image characterization, as you just saw from the illustration I showed you just before. The other thing, and to finish on that, is the plate is a 96 well plate format. That means it's fully compatible with any lab equipment. If you're talking about microscopes, if you're talking about fluid handling, automation, whatever, this is a fully automated possibility with our devices. And that's what we're doing, by the way, in our laboratory. We seed and we also feed our devices using Beckman-Coulter, for example, handling robots. Okay? So that's the 2D architecture as it is. So you remember, you can do co-culture, tree culture, and then you have projection of neurites that goes from where they are introduced in the channel here into the other channels. Okay. So we have 3 different architecture so far for the 2D devices. So we can do co-culture. We can do co-culture with a more off-centered middle channel that helps for synaptic isolation and studite. For example, if you have nerve injury and axotomy, then you can study much better the effect of how to recover using a drug for this recovery step. The third architecture is a tree culture possibility. So that means that the middle channel is as big, as large as the two other ones, okay? All right, and that's just the way we propose the plates. Half plate, which means 8 chips, or 16 chips in the full version. Okay, now what's the readout you can obtain from our devices? As I mentioned, you have a lot of imaging capabilities, and you can do immunofluorescence, you can do calcium imaging, you can do, live data assays. That's absolutely possible. And then you have the possibility also to collect the supernatant into the wells. That means that the analysis of the supernatant can be easily made during the drug testing, before or after, whenever you need it. So that's something that is largely used by our customers. And finally, we will talk about that in a few minutes, but there are also the possibility to measure the electrical activity of the neuronal network using electrophysiology, what we call MEA, okay? And we will talk a bit more about tier measurement also for the 3D architecture in a second. So just give you some example here of characterization you can make through microscopy. For example, here using paclitaxel, which is a very famous molecule for CIPN, where applications where you can see the effect of the concentration of paclitaxel versus the development or the retrieve of the neuronal activities. And then you can measure that through breadth or surface coverage. Okay, and the same thing here if you use oxaliplatin, which is another molecule used very much in oncology treatments. The other architecture that I would like to talk to you about is the 3D architecture, and this is called this is our name, our commercial name. It's called duplex. And basically what it means is that you have, in a sense, duplex apartment means that you have 2 levels. You have to have the upper level and then the lower level. And that's what we are talking about. The upper level will be the central well, where you can put a 3D biological material, whatever, if it's a biopsy, if it's a organoid construction, an organoid, for example. And then the side wells here are connected together, and they form with the beneath chamber the possibility of doing endothelial cell, circulation or immunocirculation, blood circulation. And the separation is made by a porous membrane here that does allow only the molecules to go through, but not the fluids, into the central well. Then you have here some examples of the effect of, with and without endothelial cells, for example, into here keratinocyte, which is cells from the skin, and see, for example, without and with, here, if you take the green one, where you see the density and the morphology of the pore size that has changed when you have endothelial cells that are working. Just let you know that this has been done on a static mode without any rocker at this stage. So there are other applications where you would need, shear stress also. This is another example. Here it's organoid, cerebral organoids from cortex, where you basically conducted study of 21 organoids that we grow, what we have grown into our device, which is the green a color here, versus a P24 plate, conventional plate, which is the blue one. And what really is striking the eyes is the capacity in all devices to have a very regular and repeatable organoid growth, at least for the brain one, so the cortical one. So compared to the blue one, which is the conventional plates. The reason for that is because we used to change the media from the side well, which allows the organoid to be not to be stressed, and conducting quite well the growth as expected. So it opens a large scale of studies for brain organoids, using comparing healthy and patient data organoids, basically, and so to be able to compare those with a robust process. Then moving forward, we have a roadmap for a product that will allow us to go into a multi-organ. And the way we do it is basically we're going to connect the beneath chamber here, which represent the vascularization flow. We're going to connect them from one chip to another chip. And the way to connect it is to put some bridge, what we call jumper, here and here, to create a recirculation, okay, of the same vascularization flow within the two chips, okay? And that's the way we do it. You can see here the jumpers that are materialized on this picture. And of course, in this case, we obviously need dynamic 3D. And this will be done through a little perfuser, which basically adapt perfectly to the on top of the plate. So again, that's a very simple way to integrate multi-organ into our technology. So this is not yet available, but this will be soon available. We have already tested it with the water, but of course we need to validate some platform, biological platform, using normal cells and regular cells. So these are the different architecture you can get into 3D. So as I mentioned, the one where I showed you endothelial cells with and without keratinocytes, that's the duplex typical one. And the duplex link is an additional channel here, lateral channel, where you remember those micro channels technology that we have for 2D. Here it will be combined with the 3D central well, and we will innovate basically. And we have already preliminary data already in-house where we show the sensory neurons, for example, to go and cross and innovate into the central well. So vascularization and innervation here, and only vascularization for the duplex. Again, the format for availability is half plate, so it is 8 chips, or it will be 16 chips. Again, it's going to be a 96 well plate format as before. Okay, now let's talk a bit more now about how you can benefit from NET3 technology using neurons and growing neurons and using them as a sensor. As you know, the neurons have this great capability in our body to feel, to analyze, and to communicate with our brain and back to the organs. So what we have developed at NET3 is fully implementable and optional, which means you can use with or without those electrodes, the possibility on the same chip to have, if you decide, the option of micro electrode arrays. That's the black spot here you see here. And this is about up to 48 electrodes that are mapping the region of interest where the basically the cells are growing and interacting each other through, for example, the neurons, which are crossing each other. So it means that we will use the neuron as a sensor of the sensation of the pain, for example, and objecting the pain. Okay. So this is something that we can do in our devices when we add a sheet here at the bottom of our current device, which I presented to you. So it's, as I said, it's an option. And when it, of course, it is bonded, then you cannot remove it. But this is optional from the beginning. And after that, what we do is all displayed together now fits into a measurement system, which is an Axion biosystem. The model is called Maestro, and it's a plug-in and plug-out. That means that you put your plate into the system, then they will analyze, collect data for 10 minutes. It's also an incubator, this Axion biosystem, and then you can put it back, for example, in the incubator if you are trying to find if there is an electrical signal during the maturity process of the cells. So the Axion biosystem is mandatory for our devices, as we don't have any other compatibility so far in the market with other suppliers. So here, again, you will get the same type of architecture that you got for the non-electrophysiology, non-MEA devices. So 2 channels for co-culture. Again, 2 channels for co-culture, but again, off-centered, meaning that you have synaptic isolation here, capabilities. And finally, the tree culture that is possible using MEA technology. Okay. That's the half plate, full plate, as you know now. Okay, so now we have some examples here of, moving forward. That's a roadmap over the next 3 years, how we want the multi-organ platform to be addressed in our technology. As I said, you will have one technology, one chip here with the same chip here, and then we will make a flow of the vascularization, channel. Okay. So you can address multi-organ for oncology, for gut, for any toxicology in osteodyes using different organs. That's the goal. That's the project. Now, if I come back to the 2D and to neurons as a sensor, you would see that here's some example, of course, nice images, but also if you introduce a compound into the neuronal network, then you will see that there will be a certain time of first excitation and then a resting time before recovery and coming back to the initial states from each part. Okay, so here's some examples that we have also for the CNS applications that you can be interested about, and we have a publication and also development with hospitals, and this is one of them here. By the way, you will see, or later on I can show you that you can address all the publication and all resources that we have published into directly from our internet site. There is another type of, I would say, architecture that we are working on at this stage, is the ability to have a 3D model with multi-electrode array, with electrophysiology measurement. This is a bit early at this stage, but we plan within a year to launch this product also in the market. So we can, of course, measure the electrophysiology and the electrical activity of the neurons different ways. One of the way is to use what we call slow acting response time, which means that you will introduce the drug testing in your laminar flow, and then you will put it into the reading system, the Axion biosystem, and you will measure the electrical activities. So that would be one way. The other way would be that you measure directly the compound X you are testing on the machine, so it's kind of in live measurement. Okay. So both techniques are good, and this slow acting techniques allow you to do chronicity studies, for example. So you can do one recording and then come back 1 hour after and do a second recording on the same chip and testing the same drug or different concentration of drug based on the questions that you would like to answer. Okay. So these are, different examples of molecules that we have used. I talked to you about paclitaxel, and we are also using capsaicin, and we have also neuroprotectors like propionates and capsaicin that are used, and where you can see the effect through the electrophysiology measurements and what we call the mean firing rate, which is the spiking of the electrical response of the neurons. And you can see also the effect, for example, here of a KCL application on cells that have previously seen the propionate or not seen the propionate. So you can see the difference that, for example, here, the KCL does not have any effect on cells that have the propionate because they are basically protected. So we have also ways to control make quality control of our devices and measure the viability of our cells before drug testing, and thanks to the electrophysiology, because it is a non-destructive method. This is very important. So if we, for example, if we raise the temperature, then you would see that you would get excitation of the network of neurons. And so we are capable to measure the MFR. And then when the temperature comes back to the level that we want, which is 37 degrees back, then we will see that normally the line of recovery should match the initial line. That helps a lot because that's a quality control, simple to do. Of course, at the condition you monitor well the expertise of electrophysiology, and that helps us to have, as we do services in our laboratory, that helps us to check the cell viability through electrical activities before testing drugs. So here you have some examples of different, metrics that we can use for looking after the electrophysiology readouts. Okay, and basically at the end, you will be able to compare the digital signature of altered states versus a reference state. So really, the idea for us is we are not in absolute value, we are in relative value. So we always compare a reference state, a reference compound versus a compound that will alter potentially the cells. So that way we always measure, we make an average of the differences, which is much more consistent with what we're looking for. Okay, so here again showing, early stage of standard metrics, and also the different, parameters that we are looking for: number of spikes, number of active electrodes, etc., etc. So we have about 6 parameters now we are working on to allow to pave the digital signature into our devices and into the and basically to object the sensation. So that's very important. That's basically a very extensive work that we are conducting now. It's going to take another year. It's very long, but we are gathering data at the same time and using this technology. The idea at the end is to propose to the people, to the users, to the customers, that if they have a drug in mind, and if this testing of the drug is comparable to one of the signature that we have already in our data bank, then we're capable to foresee and basically forecast the sensation of this compound that they are looking for. So as a summary, you see that we have quite a very easy to use technology. As I said, no pumps, it's a standalone device. The other thing is we are capable to replicate the compartmentalization of the anatomy, which means that we can address through neurons a lot of organs, innervated organs. And that's why we're going to talk a bit more about the applications now. And finally, the compatibility with the lab equipment using our technology is simply a magic. So that's really an important step ahead. So one more time, the neurons can act as a biodigital sensors, and that's really what we want to do. We want to address as much as possible any type of organs. Of course, there are some organs which are more complex than others, but that's the idea behind. And finally, to build this digital library and basically to have this digital signature to predict models and the efficacy and the toxicity of the drugs. So that's just a summary of the capabilities. If you want to use the technology of NET3, you can either buy directly the devices on your own, and then we will, with all support, we will provide you, guidelines how to, with the, sound protocols, and also readouts, but you will conduct yourself the experiment in your lab. The other possibility is to conduct, experiments in our lab, and that's what we call the screen. It can be large screening or small screening or, different type of project size that is possible. And we work, as of now, we work in the skin and dermatology field, as well as in the biopharma and the nutrition. And finally, we have for some big customers, we have already co-development of projects that helps basically to tailor very, very specifically project with our customers. And that's something we do in our labs. And sometimes it's a common work between our labs and the lab of the customer as well. So let me briefly talk about the corporate positioning a bit more. So really, as I told you earlier on, there are, hurdles and people are struggling, from the discovery of new molecules to get them into the process as it exists today into an FDA or EMA approval or JPA. The thing is that the preclinical step as of now is a lot done by using animal models, which give us quite some challenges because there is no good translationality between preclinical and clinical stage. That means that we want to do in NETRI is to use already human cells and HPSC, for example, derived cells that we can conduct directly from the preclinical step. That's really what we are doing, and that's how we want to increase and get faster into this direct discovery process. So finally, we want to reduce, the time to market and of course, reduce the cost of the development of a new molecule. So that's the market in 2022. And that includes, the CRO in vitro here. We have a $6 billion business, generally speaking, as a market. And it's quite fairly distributed and split between US, Europe, and Asia. Within this, there are the organ-on-chip market, where the people are using either services or the devices, and that's a total of 80 million euros, quite modest at this stage. As I told you, the technology is new. It has less than 10 years ago, and there is still quite some slow adoption with the users, but they are, of course, early adopters. And also one of the things is that we will talk a bit more later on in this presentation, the regulators are also a bit slow, although there is the FDA 2.0, which is pushing a lot the organ-on-chip into the the picture, basically. So there are basically some great projection of the organ-on-chip market for the next 5 years, actually next 4 years now. And the idea is to reach for this market, and that's again projection from Allied Market Research, about $1.6 billion at the end of 2029. So in 2030, basically, there should be a much larger adoption of organ-on-chip. And we see it as a supplier, we see a lot of momentum as we speak, using our technology. Of course, great excitement, but also people that really projecting themselves using preclinical processes with organ-on-chip. And we plan to be in the 10% market share at the horizon of 2030. Of course, there are also still some market growing over the next 5 years to 10 years from now, but it's a bit, uncertain where we will be and where the market will be. So let's keep it, a bit apart. So the real idea is the potential of the technology at NETRI is really to address a lot of different applications. As we speak, we are capable to address neurological, NDD as we call disorders. We address already the pain and injury, we address the skin and dermatology field, and we're working on the gut. Of course, there are other applications where customers are willing to do and test NETRI already, for example, in the ophthalmic, but also in the respiratory field, and also in a neuromuscular junction, which is also a big topic at this stage. So, the application is as it is as of now, but it's going to change over the next years and enlarge dramatically. So why, basically, in NETRI technology is a bit incomparable? Basically, you have two aspects of measuring the benefit of using an organ on chip. Number one is your capacity to replicate several times the same results and have a robust scientific conclusion. That's number one. And the second thing is how you are capable to be as close as possible as the function of an organ. And here, the biology digitalization is a very important step moving forward, because we have nerves all over our body. Whatever, if it's PNS or CNS, we do have nerves, and they are the element that is sensing. So you need to have the technology for sensing and objecting the sensation, and you need also to have not an academic only solution. You need to have a robust and industrial capabilities for having a high throughput. And this is what we're proposing. And as you understood, there are many actors in the market. Most of them are in this region where they have a low throughput, okay? Some of them are in the high throughput, but they don't have this capacity to do the biology digitalization. And then if we put a 3D, axis into this picture, then you would talk about easiness to use. So what is the threshold of adoption of this technology? And we would be far higher than the others. That's probably why also the customers like us so much on that. So again, it is very important to integrate the technology into your questioning, into your studies, and having the right technology will help you to respond, to answer your questions. Not the right technology, you will be stuck at some point. If, for example, you find a nice model with a low throughput, but then you have just a proof of concept and you would like to go much higher and develop your study and having a robust conclusion to develop your drugs, then you would need a high throughput. So you need to think about all those stuffs. So in terms of how we are right now addressing the market, as I said earlier on, we are already selling the hardware since the end of 2022, especially for the 2D architecture. Now, since June 2024, so only 2 months ago, we have introduced the 3D architecture, which allows vascularization. And soon, before the end of this year, we will introduce vascularization and innovation for 3D, which will be a leapfrog compared to what the market is having at this stage. The other thing is of course, as I say, we have laboratories and we are basically capable to do the services in our laboratories, building platform and testing drugs. There is also the possibility for now we're entering into the CRO partnership where the CROs are looking for using our technology, using our model, and that's basically what we call share the models. So there are other things that I told you that we are working on at this stage, which is populating the digital library with the digital signatures that we are basically constructing, and that will help us to address several things in the diagnostic world, but also in the personalized medicine, as well as in the quality control. I'm just checking the time. I want to give you some time. So it's 42. So I'm going to go very quickly then into that. So that's basically to summarize where we are is the ability to do neuron as a sensor. And that's something you can do with MEA or without MEA. And then you can use neurons and supernatant, for example. And then you can use co-culture depending on the technology you're looking for. But co-culture is something that the customer likes a lot. Okay, so this is the services. I'm going to go very quickly. Just for you to know that in terms of initiatives, we are working with many institutions and organizations worldwide. Just one example, because we're a French company, and then we have already what we call organ on chip branch, institutional branch, where we're working with basically either academia or with industrial partners. That's basically how I would like to close my talk here. So I'm sure that there could be some questions at this stage, possibly.
Yeah, thank you, Thierry. Any questions for him? Let me begin with one question, perhaps.
Sure.
So while I'm not a practitioner of science anymore, we encourage the adoption of these technologies in India, particularly because India also has a regulation in place, which is very similar to the FDA Modernization Act. It allows the use of these technologies in the Indian ecosystem now. So as a company that is raising such technologies, how do you encourage sponsors to adopt them? Is it the sponsors come to you requesting your services, or do you actively engage with them, convincing them of the power of these models?
Yeah, I think the best answer is it's both ways. Actually, we are present into the some of the decision centers worldwide. If we talk about the FDA, for example, or if we talk about the European Medicine Agency, for example, we have already discussions, and we are also part of some of those thinking, workshops. In sometimes some members of the board from our scientific team, for example, talking about the FDA. FDA is already using since 2 years of technology, and one of the key things for them is to talk about safety. For them, we can see definitely that FDA is pushing a lot about safety, and they are conducting studies using the NETRI organ on chip. So I think they are trying to build data and confidence using organ on chip before to basically emit some recommendation, if not obligation. That's number one. Now we have also the industrial partners, industrial players, like the big pharma that are also, if you take for example Roche, which is one of our customers, but there are many other big pharma which are customers. Roche is a very early adopter for the organ on chip. They have put a lot of money into a dedicated center for that in Switzerland, and we work with them. And basically what they are, I think at the end was the idea for them is to introduce and to use the organ on chip within their process of drug, of drug discovery. That way that they will submit it then to the sponsor. They will submit it to the authorities and say, listen, I have used this technology to make those results, and I need you to basically validate that. And that will be basically, we don't know which one will start first, but I expect, because I am in the pharmaceutical industry since about 20 years, there will be some icebreaker from the industrial pharmaceutical, more specifically, I think, or possibly a big cosmetic company, could be also, and they will force, they will push the regulators to change their mind on that if they are a bit slow. But as I said, the regulatory institutions, they are already working, and they are really thinking about organ on chip. And I would say that the animal model, as we know today, is going to largely decrease. And that's the sense of history. It's probably the same in India. It's the same on the Western countries. There is a value not to use animals anymore, and trying to be efficient and ethically wise, also to work on human cells. So that's going to be the sense of history. We have, there are also markets, additionally to what I said, which are already forbidding the use of animals. Like if you talk the cosmetics, it happens in the early 2010 years. Now we have the nutrition. It's in 2030 in the Western countries, and especially in Europe, no more animals. And then the pharma is a bit behind, because also it's much larger, and you touch the human human health. So it will be a bit more complex. There will be also some parts that will allow organ-on-chip, some others will not. Very sensitive one will come later. You know, this will come, but that's basically where we are in. The only thing I can tell you, Yusuf, is I joined Netri a year ago. I started to travel a lot worldwide and act worldwide. Compared to the last quarter, 2023 to now, I see the same people. I'm going to the same conferences, but I see much better, much more maturity in not what is organ-on-chip, but how can I use organ-on-chip, which is a big difference. And also the level of question, the level of knowledge is really much more mature.
Thank you. That was very insightful. Any other questions? I have another question about standardization. We've been discussing that as well. And I know that there have been recently a lot of publications or private documents from agencies in the Europe and US, which talk about standards that each of these technologies must adhere to. So are organizations like Netri a part of these discussions? What do you feel when it comes to standardization? Does it curb innovation in some aspect, or is it something that's going to push the field further to make it more widely accepted?
I think it's a very good question, Yusuf. The idea and that, you know, the goal of standard. Standardization is to lower the threshold, implement those technology for a benefit of the human health. So in terms of, you know, how we are acting with those organizations, I think I have one guy in my team. He's called Benoit Maisonneuve. Is really the guy that is, you know, moving into all those organizations and working with them. So yes, we have already some discussions, preliminary discussions, but I think it will go further and further into this topic of standardization. That's number one. So you have to be present with those organizations and understand what they want, and also see if your current technology is already addressing most or a part of the request. The second thing is that in terms of you know, innovation, standardization here for us means that it's not necessarily correlated with innovation. We think that the technology that we have is a very easy to use technology. So standardization means several things. It could mean easiness to use. It could mean capacity of delivering the devices, which is another thing, which is linked to complexity in a sense. And basically on that respect, there is only a very, very, very limited number of players worldwide that are capable to already say, I can use my technology and standardize it. We believe at Netri that we are the closest. We're not saying we are the one, we're saying that we are the closest. And standardization means, for example, automation. So if this become, if the standardization means that you will use organ-on-chip not only for drug discovery, but also for things like, you know, quality control, then you will need to have to deliver throughput on that. And that's also a very, very limited number of players worldwide that are capable to do it. Actually, I don't think about two, if not three worldwide that are capable today to say, I can be the technology of standardization. We are definitely one of them, but I'm not going to speak for the other ones. But that standardization is a key point. You address it correctly, Yusuf.
Okay. Thank you. Thank you very much, Thierry. We'll share your contacts with our participants. If they have any further questions, they'll get back. I'll also share the presentation. And I've also collaborated with Benoit earlier for various academic reasons, but it would be wonderful if we could all team up together and do something productive in the future. So thank you.
Yeah, thank you very much, Yusuf, for your invitation. And as I said, Netri is a very agile organization. We are very close. We are, as we say, we are a human-sized company. So we are here to respond, and don't hesitate to solicitate us. And that's going to be very appreciated from our side also.
Thank you.
Thank you very much. Thank you.